Okay, so we're going to talk about the structure and function of the plasma membrane. Remember, the plasma membrane acts as a selective barrier that separates the intracellular fluid of the cell from the extracellular fluid. It plays a dynamic role in cell activity by controlling what enters and leaves. That's why we say it's dynamic, because it's changeable. And we also call this the cell membrane. Now, what we see in this picture is basically a side view of the plasma membrane or cell membrane. And this is if you're zoomed in on it a lot. So this would be the extracellular fluid, kind of a watery environment outside the cell. And this would all be the intracellular fluid, including cytoplasm. So what we see here is the plasma membrane itself, and it's not drawn to scale. I mean, it's zoomed in quite a bit. And what you find is actually a lipid bilayer. So each one of these little circles here with the chains coming off is what we call phospholipid. This end of the phospholipid is polar and therefore attracted with water. These ends are the nonpolar fatty acid tails, which are hydrophobic and therefore are water-fearing, so they actually hide from water. And what you find is that the plasma membrane is a phospholipid bilayer, which means you have two layers of phospholipids that point at each other, where the phospholipid tails here, the fatty acid tails, point inward, and then the polar head groups are the ones that interact with water in the extracellular side here in the outer leaflet, or this side, and the intracellular side here in the inner leaflet, which would be this side. Now, what we find is that there are some other lipids in the plasma membrane as well, not just phospholipids, but also cholesterol. So cholesterol plays a really key component in terms of membrane stability. So each of these little gold things here is a cholesterol molecule that actually helps to stabilize the plasma membrane. You find that there's also a lot of protein that's embedded and associated with the plasma membrane. Some of these are involved with things like transport or cell anchoring, enzymes. Some are involved with communication, and they play a variety of roles. We also find, too, that there's things called glycolipids that are attached. And a glycolipid is essentially a lipid with a large sugar molecule attached. These are also involved with cell signaling and attachment. Same with glycoproteins, except for this, the sugary part here, the glyco component, is to attach to a protein that's embedded within the membrane rather than a lipid itself. So in terms of just general functions of the plasma membrane, first of all, it serves as a mechanical barrier. And that makes sense, because mechanically it separates the two fluid environments of a cell, extracellular and intracellular environments. Secondly, it plays a role in selective permeability, so it actually aids in the transportation of certain molecules across the membrane, but it's selective in the sense that it allows some things to cross and other things not to cross. The membrane also serves as an electrochemical gradient. So what we find then is that there's a separation of charges across the plasma membrane, where you'd find that the inside of the cell could be slightly more negative than the outside. And because of this separation in charge, it functions like a battery. There's actually stored potential energy here between the extracellular fluid and intracellular fluid. And what's cool is that muscles and neurons can use this for communication around the body as well as muscle contraction. Now, the plasma membrane also plays a role in communication because it allows for cell-to-cell recognition. That's where the glycolipids and glycoproteins would come into play, because these are like the name tags of your cell. And then it also plays a role in cell signaling, where membrane proteins can interact with different chemical messengers in the sense that they can act as a receptor to relay messages to the cell interior. So in terms of just the structure of the membrane, remember, most of it's made of phospholipids. So 75% of the lipids of your membrane are phospholipids. And there's two major parts. We have the hydrophilic and hydrophobic regions. Remember, the phosphate head groups, which are polar, are the ones that are hydrophilic. And this is why they're associated with the watery extracellular and intracellular fluid environments. The fatty acid tails are nonpolar and therefore hydrophobic, and that's why they actually don't interact with water and they protrude inward towards each other. Now, 5% of the membrane lipids are glycolipids, and these are the ones with sugar groups attached. These are often involved with attachment and cell signaling. And then 20% of the membrane lipids are actually cholesterol. So a pretty decent amount of those lipids are cholesterol, which actually increases the stability of your plasma membranes around your cells. Now, in terms of membrane proteins, they have a variety of functions. Some allow for communication with the external environment or the internal environment. Some make up about half of the membrane mass, and most of these have specialized functions. Some float freely, others are tethered, and there's two major types, integral and peripheral proteins. The integral plasma membrane proteins are the ones that span through the whole width of the membrane. They go all the way through. And the peripheral proteins are the ones that are just sort of associated off to the side. So what this slide is showing are just all the different functions of plasma membrane proteins. So some are involved with transport. We can find that certain transport proteins in the membrane can help to move molecules either out of or into a cell. Some can either be passive or active. If they're passive, they don't require ATP. If they're active, then they do require ATP to move a molecule either out or into the cell. Some of these are also involved with receptors. So some plasma membrane proteins are just receptors for different chemical signals. And they're involved with transduction, which means they actually can help relay the signal into the cell in some regard. Some of these plasma membrane proteins are also involved with attachment. So for this integral membrane protein, you can see that's actually attached to the extracellular matrix to help keep that cell in place and in position within a tissue of your body. Some membrane proteins are enzymes in the sense that they can help with metabolism or even cell communication. Some membrane proteins are involved with intercellular joining, where basically this is one membrane of a cell here, this is the membrane of a different cell here, and you can see that these two cells are really close together because these plasma membranes, the proteins are actually interlocked and interjoined. We call these cell adhesion molecules, and they actually help for binding between different cells. Now, cell-to-cell recognition can also be used for things like immune cells, where the glycoproteins on one cell act as a name tag, and then we have a receptor on a different cell here that can actually, pick up that name tag and understand whether it's a cell of your own body or if it's a cell of a different organism, or like a foreign cell, you might say. Now, the cell junctions are basically just different types of proteins that hold cells together. Some cells in your body are free in the sense that they aren't bound to other cells. Examples of this would be like blood or sperm cells and some immune cells. They're not necessarily bound to other cells, so they freely float around the body. But most cells are bound together, especially the ones that form tissues and organs. The three ways that cells can be bound to each other are things like tight junctions, desmosomes, and gap junctions. The tight junctions essentially hold plasma membranes together, and they help to essentially waterproof tissues, so they prevent water from passing between cells. The desmosomes here are a larger type of membrane protein that really tightly hold cells together, and they also interlock with the cytoskeleton inside of those cells. And then gap junctions are involved with cell-to-cell communication because they're effectively a protein tube. So looking at this picture here, we see our tight junctions. And the tight junctions here form an impermeable barrier where they almost look like rivets. And so what we can see here then are essentially proteins from one cell to another, and they can interlock to hold these membranes of different cells really tightly together. That way that water can't pass between these cells. In places you might want to find a lot of tight junctions are tissues that need to be waterproof, like your skin and some of the mucous membranes around your body as well. Now, the desmosomes are a larger type of protein. They're found a little bit closer to the basal surface, or kind of deeper here in the cells. And these are the ones that interlock with each other, but also are linked up with the internal cytoskeletons of the cells. And they form these large aggregations of protein, which we call plaques, because underneath the microscope, they look like these kind of larger, dark plaques on these cells. But essentially what they are large proteins that can interlock these two cells together, and because they're linked up with the internal cytoskeletons, they can really tightly hold these cells together well. Now, the gap junctions, you don't find these in all cells of the body, just certain cells that have specialized functions, especially for communication of like electrical and chemical signals, as well as passing of nutrients from cell to cell. Because effectively what these gap junctions are a protein tube. So imagine you have one half of the tube on this plasma membrane and one half of the tube over here, and when they match up, they form a tube that connects the intracellular environment of one cell to the next. That way some molecules, if they're small enough, can pass between cells from cell to cell. And you find gap junctions, in embryonic cells as well as heart cells for conduction of electrical impulses from cell to cell. Now, this kind of leads us into our next question. If the plasma membrane is involved with transport, the question here then is, how do substances move across the plasma membrane? Especially if they're the ones that are nonpolar or polar, how does the membrane differentiate between molecules that can or can't cross? Because the membrane has selective permeability, so some molecules can pass through easily, some can't. And there's really two principal ways that substances can cross. We have passive processes that don't require any additional energy input, so it doesn't require any ATP to transport certain molecules across the membrane. Otherwise, we also have active processes that do require ATP. And that's why it's called active transport, because they actively require the use of ATP to move a molecule from one side of the cell to the next, like from the intracellular environment to the extracellular environment, or vice versa. And if it requires ATP, we say it's an active process.